Thank you for the wonderful, kind introduction. My conflict of interest that I'm the site PI for GLM 101, and I was the main PI for the Epalrestat study. I don't really need to talk too much about CDGs in general, but if you look at these features, developmental delay, speech delay, gait problems, tremors, seizures, affect most of our kiddos and adults with CDG. There are a few disorders which are even progressive in nature. We have no FDA-approved treatment for CDG. This is an unmet need. We have to do something about this as soon as possible. Today, I will show you a couple of examples of treatments which started from N of 1 trials, just to give you a little taste of it. And Sunday, we will go a little bit more deeper in details and maybe answer more questions. I start with a very fun example of how a series of N of 1 trials can happen. I start with PGM1 CDG. PGM1 CDG has actually a problem here. And even though it's linking two different glucose molecules together, it affects galactose or milk sugar metabolism as well. And these patients have a lot of muscle symptoms. They also have other problems and hypoglycemia or heart problems, but they have significant muscle problems. And so one of our kiddos has a very young trainee of mine who I trained at NAMA. And one day I was called by this trainee who is now a metabolic doctor, and he said, I saw a paper in the literature about something really interesting in PGM1, and he sent me the link. So this paper is in Chinese. So I tried to read it, but I couldn't, but I had Chinese colleagues who helped me. And the bottom line is that actually these physicians found in one single PGM1 patient that the muscular and neuromuscular junction in the muscle didn't work well. So you have to know that when your nerves are giving orders to your muscles, there is acetylcholine, which is released and then gives the information to the muscle. And in this patient, they found a severe abnormality of this transfer of information between muscle and nerve. So actually, this seems like there is a new aspect of the disease we didn't know of. And so my dear colleague started his patient who had muscle symptoms, no myasthenia, like in this patient, but muscle symptoms, muscle pain, muscle weakness. And guess what? The patient really improved in her muscle function. So actually, since this case, just vignette, there are 3 other patients with PGM1 who are on this drug, which is an off-label use of this medication. And the whole series of the trial series is based on somebody who just open-mindedly read the literature and found something interesting which was relevant for our patients. So this is a great story because actually all the 4 patients are doing really well, full of energy and less of muscle pain. None of the 4 patients had rhabdomyolysis, a painful muscle problem since they are on this treatment. I bring in also another example for how you can use N of 1 trials to develop future treatments in CDG. In this case, we go to a more a complex pathway. You see here their fat synthesis happening here, which is going to a very special fat, which is carrying the sugar molecules for glycosylation. And the defect is here. It's called DHDDS, CDG. And patients with this disease have a really complex neurological phenotype. When you see here, you see all the different neurological problems you can experience in this disease. And you can see Rosie, who offered this video to share with you, how difficult for her to walk the line, and the toe to heal walking is significantly difficult, showing ataxia and some Parkinsonism in the type of like a slowness of the movement. So this is a disease which was discovered only less than 10 years ago. It's an autosomal dominant form of CDG, very rare, and there is a relatively common mutation. So Ethan Perstein and his colleagues, Christine and the team at Perlara, developed a yeast model for the disease. They did drug screening, and when they gave these 8,500 compounds, I think, to different yeast, so they grew enormous amount of yeast in the dish, and then sprinkled different drugs on them, and then they looked, okay, this yeast didn't make it. This yeast got even worse. This yeast, there is no one there. And then there was something they gave to the yeast which made the yeast survive and grow. So then they said, okay, so what is this mysterious drug? And so when you look at this chart, sometimes it's a little complicated to see all these tiny letters, but you can see that these are the compounds and how they help. The higher you've been, the better helping that compound your disease. And you see here the controls, and you see the disease yeast. So you see some of these compounds are really improving function, not fully restoring, but really improving. So what was this mysterious compound? This was actually something we call NAD precursors. These are molecules which are important in the vitamin B2 metabolism and well known to help mitochondrial function. So then we think, okay, welcome vitamin, we love vitamins, but like how would that help a CDG? So Thomas and Irena developed an organoid model in our lab. We heard about organoids in the first session. You see fibroblasts, they were reprogrammed to pluripotent stem cells, so they were like embryonic, and then we made neurological cells from them, and they developed to organoids. We also learned we cannot call them mini brains, but that's how they look, tiny little brains. And then you can harvest them and evaluate them. So you can measure glycosylation, you can measure their function, you put like an EEG kind of thing on them, and you can do expression analysis. So it's like an amazing source of human tissue in an embryologic phase of neuronal development. It doesn't solve all our problems, but it's better than a brain biopsy. So you see, we did a lot of studies, and I don't really have time to go through all that because I'd like to focus on N-of-1 trials today. But what you see in this graph, that there are differences, big differences between bars. And if you look at those differences, the biggest difference is like, the patients are really lower than the controls, but also you see between the green bars, you see that there is a significant improvement of basal OCR and maximum OCR. And Thomas taught you about energy metabolism and how in seahorse we can measure these energy production parameters. And you can see here, wow, this is really an obvious positive effect on energy metabolism. So well done. What should we do as the next step? Our Cure DHDDS Foundation supported the notion to start some patients on this supplement, which is a form of vitamin B2 or niacin. So it's well known that it's safe and it has restricted side effects. Maybe in some forms it can cause flushes, or it can, in some of the forms can increase problems to fall asleep, but otherwise not dangerous. So patients started on different doses, and we saw that higher doses actually didn't work better than the lowest dose. Highest doses actually caused more side effects like irritability and sleeping problems. And so what we decided that we actually try to have everybody on the same protocol, 250 milligrams of this molecule NMN, and then we just collect the data on the patients through the natural history study and see how the patients perform. And what we do is a test called Icahn. So we use this test, Icahn, to try to have a baseline, and then we treated the patients and then repeated the Icahn. On the first video, you see Rosie doing and drawing her Archimedes spiral. And so if I restart this video, then you will appreciate even more how much more fast and smooth she's drawing. Maybe I don't know how to go back, but you see like the perfection of drawing the spiral. That's really very subjective, yeah? I just picked the best video I got from her. Can we actually measure that her function improved on the drug? So we decided first to try to measure what is a normal motor pattern or gait pattern. And what you see here is a healthy individual walking, doing that toe to heel to toe walking. And when you see him walking, then you also see this graph next to him, and you see where are the ankles and the wrists compared to the midline. Whew. I don't know what happened. Now I'm sending SOS. Yeah, I'm sorry. So now somehow we went back or went forward. Yes, so just look at him. I don't know why the video doesn't play anymore. Sir, can you help me and play my video? So you see now Rosie again, and her movements, and you see this graph, how far away are her arms from the midline. And then this is a video after 4 months or 6 months of treatment, and you can see the hand movements and the feet movements, how much closer she got to the midline. So what we decided, we need better way of, could you please get my presentation back? So what we decided is to try to make an objective measurement of these movement abnormalities. And here you see some of the graphs we tried to statistically evaluate. One was Icahn in several patients, which improved significantly. This is open label, so it's it could be misleading because we are optimistic and we score very optimistically. However, this is a progressive disease, so you would expect actually worsening, not improvement. And you see also here improving jerks and movement abnormalities. So I will come back to this in my clinical talk on Sunday. But right now, the point I wanted to make is that single observations could be bundled as a series of N of 1 trials. If the different clinicians are following the same protocol, you can actually collect information based on these outcomes. And right now we are starting a clinical trial for DHDDS CDG, which got just IRB approved on Monday. And we also have an IND from the FDA for this trial. I'm generally an optimistic girl, and I think you are waiting for these results for a long time. I would like to come back to the Epalrestat trial and share with you some of the observations we made during the trial. So not everybody knows the concept why Did we use an aldose reductase inhibitor in PMM2 CDG in a large group of PMM2 CDG patients? It also started with Perlara and Icahn, who did drug screening with the support of Maggie Sperl and Holly to establish or find a new drug for PMM2 CDG. This is a very complex pathway. I will show a simplified version on Sunday to you, but the most important part is that here is the gene defect, PMM2, and the drug Icahn found is an aldose reductase inhibitor, so it's quite far away from the defect, but you see that the sugars are interlinked, and here is sorbitol, which is a product of glucose to sorbitol pathway when aldose reductase works. And the drug we have been evaluating was an aldose reductase inhibitor. So here's the block. The consequence is that you have elevated sorbitol in fibroblasts and urine and blood. And then the idea is that if you block this pathway, you stop making sorbitol, but you also start the metabolic flux this way, so it doesn't go towards sorbitol, but it goes this way, increasing glucose 6-phosphate and glucose 1,6-biphosphate, which is an activator of the PMM2 enzyme. So we measured that it indeed goes up, and glycosylation increases based on the drug screening, preliminary studies, fibroblast studies, and a single patient IND. We went for a clinical trial. We enrolled 38 patients to the study who got randomized to Epalrestat and placebo, and the patients were further stratified according to their age as well. Outcome measures were antithrombin, which was just deducted by the observation that in our natural history study, most of our PMM2 CDG patients had significant decrease in the activity. And we thought that's a glycosylated protein. If we truly improve glycosylation, this should get restored. The other outcome measure was sorbitol, because we know that the drug, if it works, it should improve the levels. And Icahn, as I just mentioned to you, that's the ataxia or motor scale. So there was no significant difference in our patients, and it was a devastating news after going through 15 months. People traveled from all over the country to Mayo, several blood draws, a lot of testing through snow in Minnesota. I cannot describe, but most of you who were part of it remember. So the patient community gave everything into this trial, and we still failed. So what else? There was no serious side effect. That's good. But there was one patient we lost. And that patient we didn't lose him because of the trial, but because of severe multi-system disease. And there was an observation of relatively higher infection numbers in the patients who were treated on Epalrestat, and the data monitoring board worried about that too, that potentially infections could increase and could be a negative outcome. So why did we fail? Why did we fail? This is something we are working on with Icahn and my colleagues in the lab for more than a year to try to figure out. So one thing I wanted to show you that we had a large number of children with autism in our cohort. And if you look at these patients, if you see how high their ICARS is, it's maximum 100. So it means like they practically couldn't really perform the test, because when I told them, touch your nose, they didn't understand, or they just didn't want to follow, or they were unable to walk, so I couldn't check their gait. They obviously couldn't draw a spiral. So actually, you could look at how many patients we enrolled, because we wanted to be everybody welcome approach, and then we skewed our data because these children practically didn't change either in the placebo or the Epalrestat arm. So maybe statistically it doesn't matter so much, but it was just torture for the patients who couldn't really benefit, I assume, or it seemingly it couldn't benefit from the trial. So what we try to do with Lisa Kim, who's in our a new postdoc in our lab, to go and look into the data when we are not including those patients who didn't change their ICARs and had severe autism, and also some of the patients were too young to have ICARs. So you only see data here for how many patients? 30 or I'm sorry, I cannot really see from here. But so it's a number 90 or I'm sorry. These are how many patients are these? Altogether without the autism? 29. Okay, thank you. So when you compare these, it's still not significant. So that's really annoying. It seems like there's something else we messed up. So we, however, noticed that in the treatment group, there were these super responders. Like you see the Epalrestat and the placebo, how it differs. So the question was, is it possible that there are certain genetic variants which would make you amenable or make you, you know, a candidate for this therapy? So we try to see what's the difference in the genetic makeup of the patients who are responders and non-responders. And how we started this, I just wanted to show you this because Icahn Feinberg is sending his best regards and wanted to show you that he's still exercising. And after months and months, and now 3 years on Epalrestat, he made his first steps. And honestly, he's a borderline responder, and I think that his benefit is most likely more the sorbitol decrease and that his neuropathy is not worsening, but he's able to make steps, and that's really great. So, greetings from Icahn. And you see also here something interesting, how actually the baseline glycosylation marker improved in serum in 6 months in patients with PMM2 CDG on the drug. So I want to get back to, very quickly, to how we decided which patients should be good candidates for an aldose reductase inhibitor trial. Would it be Epalrestat or 007 or any new medications? So what we decided together with Lisa and one of the structural biologists at Mount Sinai, that when you use a chaperone effect, in other words, you aim at increase glucose 1,6 biphosphate to improve the activity of the sick PMM2 enzyme, then there are certain mutations which are not amenable. And these are severe loss of function mutations. Obviously, if the G16 or G16 biphosphate binding is affected by the mutation, then where would it bind? And naturally, the magnesium binding, because that needs to come together to stabilize the conformation. The linker region is very important as well. And there are some specific folding and dimerization mutations we selected. And here's a list of our variants predicting minimal treatment response. And that's mostly based on AlphaFold prediction literature of functional studies, or structural changes like proline, which is very disruptive to the structure. So we actually ruled these out without looking into the data. And when we ruled out these variants, both in the placebo and the Epalrestat, we only ended up with this handful of patients. But then you see that actually the difference is significant. So what we deducted from this experiment, that maybe we should think similar to how in cystic fibrosis or other disorders, certain drugs and chaperones could only save specific variants and specific mutations. And I think that for the future, we have to make a list of specific mutations which should be treated and trialed, and we shouldn't give up on all those reductase inhibitors. Actually, the other way around, I think we should be very excited about these, because I think there are special super responders we detected in our trial as well. So what did we do wrong? I really just want to summarize what we learned from this trial. We included all individuals across the disease severity spectrum. That was a very humane thing to do, and many people wanted to be in the trial, and we didn't want to be cruel and tell them no, but that was really silly, because the very severe patients were unable to show improvement. Maybe did improve, but we couldn't measure it. There was no open-label dose-finding study before this study because the FDA didn't let us. So the outcome measures were like this. We were just agreeing on what kind of outcome measures we should do, but we couldn't test it before the trial. But the FDA changed in that recently. I told you autism is not a good diagnosis for doing ICARs. So in the glycomine trial, we were very specific, including only patients with a certain ICAR score and with the availability for performing the tests. Antithrombin failed as an outcome measure. The last thing I said, what was also not good, sorbitol was measured several times in a non-compliant form. When where you're supposed to measure something fasting, you cannot ask your child, now you fasting now because we are in the trial. So when your child wants food, then you give food. And I think that it was a silly decision to do this instead of a feeding protocol, like we measure this and you have to drink a half glass of orange juice. Probably that's a challenge for some of us too, but the fasting is really not a good idea. I think we learned tremendous amount from this trial. I was devastated when it closed, but I think what we learned, it helps us to be really successful in the future. And some of the success of GLM-101 or the glycomine drug, I expect that is also some learning point from us, from our mistakes from the past. Okay, future perspectives, and I'm finishing up. I really want to invite you all for the Sunday talk, because our colleague Ben will show you how can we use AI methods to objectively measure movement and make a biomarker from a home video. So you see that that video, that famous video for Rosie, and you see that this is like labeled. You see these axes between wrist and shoulder and the head, and you see how she stands and how tall she stands, and you can measure the tilt. You can measure the distance between the hands and the different body parts from the midline, and the computer can learn that and do that, and the more home videos you give to the computer, the more data you collect. So in an ideal world, and that's what I'm aiming at, I am actually not dragging my patients every month to see me, but I tell them, please do some home videos as a baseline, and then I'll call you with instructions. You can start the study drug, and your PCP can take the blood, what's necessary for safety, and then we will have another home video, and the computer will assess it, so it's like objective. It's not me saying, "Whoo, you're doing so much better," but the computer say, "Hey, your ankle symmetry is getting closer to the normal." So Ben will teach you how to do this and what other magic he and Rory can do for clinical trial outcomes. And I also want to announce that we start our first clinical trial in DHDDS, CDG, which is FDA and IRB approved. We start to enroll next month. And this is a gracious support of CDG UK and the Cure DHDDS foundation, and actually FDA approved to use the home videos as outcome and decrease the number of visits in the hospital. Thank you.